Hey, Clarice, thank you for coming in. I just think your work is amazing. Just what a wonderful background in terms of the work you've done in the past and what you're doing now and what you're leading. And that's why I reached out and say, hey, let's do this interactive chat. And I should mention to the audience that just a reminder, this is unscripted, so we're just going to go where it goes. So again, thank you
Those
for
are the
coming in and
those are
sharing
the
insight.
nicest chats. The unscripted ones are the nicest chats.
So thank you for coming in and sharing your insights.
So thank you for having me. I'm honored to be talking to you and excited to share some of our ideas, some of our research. So feel free to jump right in, or should I jump right in? Shall I tell you a little bit where we're from and where we're going?
Well, what I'll do first of all is my audience is always curious. You've got this wonderful background, you're doing tremendous work, and there must have been inflection points in your life, maybe two or three. Could have been when you were really young or in school, could have been a mentor, something that happened in your career, your professional life. Name those things that were magic in your life to create this wonderful person you are today.
That's really crazy because growing up, which is very different from your usual origin story. Growing up, I wanted to be a writer, a journalist. I didn't grow up liking physics or chemistry in particular, and I read a lot though. And a cool thing is that more and more I realize how the capability to communicate my research is related to my love for reading, to my language skills that I developed in childhood. So that's a very particular point about myself. I work in a weird field called quantum biology, which is the emergent field that wants to assess the extent to which quantum mechanics is playing a role in biology natively, endogenously. And I want to talk about an inflection point that happened uh, during my first postdoc. So, for my graduate studies, I worked on technological quantum sensors as a graduate student at MIT. So, actually, one can prove that if you use a quantum object, a tiny object such as an electron, as a sensor, your measurement is improved. In other words, the sensor quantumness enhances your result. Um, After my PhD, I really didn't know what I wanted to do. I spent a year teaching in my department, and then I started sending out postdoc applications. And I was accepted to a chemistry group at Berkeley. And for that work, we were developing an imaging modality, and I was developing a tiny little chip. I was in like a nanofabrication lab all day, but I was within a chemistry group. And then our chip was designed to image chlorophyll, but the chip wasn't working with the chlorophyll. Um, we didn't know if it was a problem with the chlorophyll samples with the chip. I started looking at other molecules that we could test instead of chlorophyll. And by chance, I stumbled upon a phenomenon uh, that really looked like quantum sensing happening inside proteins. I found a class, I mean, that it was known, there's a class of proteins that respond to external stimuli in the same way that technological quantum sensors would. It's just that chemists don't call it that. So that's a pivotal moment for me because I then realized that I could maybe bring instrumentation that I developed during my graduate career to study these phenomena in biology from a very high tech level. So that what made my career change completely, and I'm like, now I know what I want to be doing, the type of research that I want to be doing. I want to be researching putative quantum phenomena in chemistry, in biology.
That's really fascinating. So you're indicating that you started with this really strong interest in literature and you read a lot, and that would really give you sort of a more interdisciplinary sort of feel, I guess. And then you end up going into the hard sciences. I mean, physics is sort of a hardcore science area, and you got your bachelor's there, and then you end up with a a master's in physics at Cambridge, Trinity College, and then you end up electrical engineering with your PhD at MIT.
Yes.
And then postdoctorate appointments in biomedical or bioengineering at Stanford, and then chemistry at Berkeley. So that you talked about how
Yes.
all of these different aspects gave you this lens to now doing this amazing work in quantum biology, where you lead the QuBiT lab at UCLA. So I can see now how all the pieces come together, but it means that you're really thinking in a lateral way as well, plus
Yes.
depth. So that's fascinating. Okay. Okay. So now to the audience, let's get into more detail about quantum biology. I mean, you sort of hinted at what quantum biology is. Well, let's get into more detail of what quantum biology is. What is quantum biology?
Okay. So, first of all, before anything and before talking about quantum biology in particular, I just want to mention that we already live in a quantum powered world, right? All this hype with quantum, like, yes, we now have quantum computing, quantum algorithms, but simple things such as lasers, magnetic resonance scanners in the hospital, GPS, even your classical laptop, they're all already powered by the laws of quantum physics, right? And I want to point out that humans developed quantum technologies after, say, 300 years after the Industrial Revolution. So I don't think it's crazy to think that nature, who's an engineer with like 8 billion years of experience, why nature shouldn't have stumbled upon it to build better technologies before. So there's evidence. All the evidence is correlative at this point. I'll explain a little bit what I mean by that. But there's evidence that nature is indeed harnessing quantumness to function and to function optimally. Um, you might have heard about a couple of the poster children of this field of quantum biology. For example, photosynthesis, right? You might know that the efficiency of the photosynthetic processes that occurs in plants, like the photon absorption and dumping of this energy of this photon, is way higher than any solar cell ever built by humankind. And one of the correlative evidence is that this is done helped by a chemical process that depends on vibrations in a quantum mechanical way. For the quantum expert that is in the audience, it's called a noise-assisted transport of energy. So photosynthesis is very famous. The other thing that is very famous are birds. There is it's been known for 50 years that birds, when they migrate north-south, that they use, at least as a partial cue, the magnetic field of the Earth, which is like orders of magnitude smaller than the magnetic field produced by your cell phone. So how might they be doing this? And nowadays, the evidence points to the fact that they do so via a quantum sensing like a phenomenon happening inside proteins inside the cells of the birds. So this is a bird's eye view of how I would define quantum biology. It's the study at different length scales and time scales of the extent to which quantum degrees of freedom native to biology are affecting macroscopic biological function, affecting how the birds migrate, how the plant processes things. I'll keep it at that to keep the surprise, because it's just keeping it to plants and birds is a gross understatement of how organism agnostic and how widespread quantum effects in biology truly seem to be.
You know, that's really, really, really fascinating. And I can see sort of this related discussion I had with Roger Penrose, where he talks about quantum gravity collapse and these microtubules, and it's an outlier concept, and that maybe this is sort of the basis of some kind of consciousness, and it's really a property of the universe or something like that. Or you see this research out of Japan many years ago where they got single-celled plasmodium, sort of like amoebas, to solve network problems by probing them with light channels. And they think, how is that possible? So something else is going on. So maybe that's has some kind of root in quantum phenomena, right? Or
Yes, and it could be. And yet, on Earth today, there is no unambiguous proof that biology is truly using quantum to function optimally. And let me go a little bit over the evidence that exists and how we're trying to improve this evidence, because that is, I think, one of the reasons why quantum biology is not more embraced by the bio community and by the quantum community. So, for example, for test tube chemical reactions for photosynthetic complexes or for other types of chemical reactions, there is no doubt that there are quantum stuff uh, happening at room temperature in a way that really changes like the macroscopic outcome of chemical reactions. Another thing that we haven't mentioned is the fact that enzymatic functioning seems to be driven by quantum tunneling. If your audience knows what that means, if not, that's fine. It's a quantum process. So in test tube chemistry, it's uncontroversial. Things inside biomolecules happen that are quantum mechanical at room temperature and for long enough so as to have a macroscopic effect. Now, the next level of evidence, it goes from uncontroversial chemistry evidence from many decades to beautiful, hard, but maybe not sufficient experiments conducted, say, by biologists at the level of like whole organisms, from birds to slime mold, as you mentioned, to plates of cells, and um, to like little organisms. And those organisms, those plates of cells, seem to respond to external stimuli in a way that is consistent to what would be expected for them to do if this type of quantumness that is found in chemical reactions were in fact responsible for what they're doing, right? Uh, all the data that exists is correlative. So it goes from chemical length scales to very, very large length scales. And I think that one of the first things that we need to do before we even think about problems, as you were mentioning, about cognition or further things, right? We need to establish the foundation of whether, say, quantum mechanical effects can be sustained inside a single cell and measured so as to make an unambiguous link or refutal that those two things, that the chemistry length scales is really driving what's happening physiologically, macroscopically.
so how are you bridging that gap? So, I mean, I read an article that you're looking at this electron quantum property of spin. So can you get into more detail of what that means to the
Yes,
audience?
that is fascinating. And I hope to convince you that this is crazy. So hear me out here. So, for those who are not familiar with spins, so spin is an intrinsic quantum property of matter. There is no classical analogy to spins, but everything has spin, like electrons have spins, atomic nuclei have spins, and spins are as fundamental as, say, mass and charge. In the same way that mass tells you how, say, an electron interacts with a gravitational field, charge tells you how an electron interacts with an electric field, spin tells you how an electron interacts with a magnetic field. In the same way that positively charged ions react to an electric field differently from a negatively charged ion or an electron, um, different spin states react differently to each magnetic field. For electrical charges, we have positive charges and negative charge, which is just a name. We could have called them like good charges, bad charges. In spin speak, we represent spin with an arrow, like arrow up represents one spin state, arrow down represents another spin state. And spin up and spin down represent different interactions, different ways of electrons, for example, interacting with external magnetic fields. Now, it's been known, again, uncontroversially for test tube reactions, that there are some chemical reactions that can be controlled via the spin property of particular electrons. Here's how this happens. the chemical reaction happening. At some point, the chemical reaction comes to a crossroads and effectively at that point looks for the quantum spin state of a particular electron. If that spin is up, arrow up, the chemical reaction continues through one branch. If that particular electron spin is down, the chemical reaction continues through another branch. Importantly, the macroscopic final processes of those two branches are different. So there is no doubt that affinity, quantum property of an electron is known to change big time the macroscopic final products of chemical reactions. Now, if at the point where this chemical reaction is at this crossroads, the almighty electron spin that we care about, it sees an external magnetic field, it's going to talk to this magnetic field. It's going to interact with this magnetic field in a way that is indistinguishable from what I was mentioning about quantum sensing. It's going to quantum sense this magnetic field, which means that this external magnetic field is going to alter the probability of us finding the spin up and the chemical reaction continuing through one pathway, or finding the spin down and the chemical reaction continuing through this other pathway. It's in this way that magnetic fields via this spin quantum knob have been known in test tube chemical reactions to alter the final products of chemical reactions. Okay? So now there's evidence that the same, that again, that's what's thought to underlie the bird stuff, but it goes way beyond birds because magnetic fields have been shown to alter a whole range of physiological phenomena in a way that is consistent with this type of electron spin dependent chemical reaction. Let me name some of those phenomena that have been observed to be tweaked to be able to be controlled by magnetic fields in a way consistent with this mechanism. It goes from yield of DNA repair to up and down regulation of cell proliferation, to control of the production of cellular oxidants and antioxidants, regulation of cellular metabolism, cellular respiration rates. You have embryogenesis being messed up by magnetic fields, epigenetics. So there's a whole lot of important things happening biologically that seem to respond to magnetic fields in a way consistent with this spin mechanism being the driving force. So that's what we want to start deterministically studying, because I maintain that once we have the deterministic code book on how to tweak chemical reactions inside cells with very precise magnetic field strengths and frequencies, we have a new knob, an electromagnetic knob to control biology. You can think about going from a chemical way of looking at therapeutics to something like an electromagnetic way of looking at therapeutics, for example.
You know, that's really, really fascinating. So what you're indicating is, and the research is indicating that you have this spin property, and that spin property controls the type of chemical reaction that can occur. And you can manipulate this using magnetic, in fact, very weak magnetic fields, right? So there's no damage, it's non-invasive, right?
Can I add this? Yes, that's a very, thank you, Stephen, for mentioning this. For reasons that are well understood chemically, so people understand why that is, the magnetic fields that can actually alter the fate of those electron spin dependent chemical reactions are relatively weak, on the order of the magnetic field of the Earth, on the order of your cell phones. This also means that if you put a vial of magnetosensitive proteins inside a big magnet inside a magnetic resonance imager in a hospital, that big magnet will not change things physiologically, at least not through this phenomenon that I'm describing. The thing here is that with very weak magnetic fields, you seem to be getting a big macroscopic biological effect. So up to now, research has been sort of phenomenological. People find sort of almost by chance magnetic fields that fit some purpose, that do something. I think that the company that really got a head start on this is a company called Novocure that is valued at almost $10 billion now. they came up with an electromagnetic device that produces weak electromagnetic radiation, and this device um, sort of decreases cellular proliferation rates for a very aggressive type of brain cancer. And in all that is published, there's no mechanistic understanding of why those fields are the right fields to do this, but it works for their particular problem, right? And now they have this device, and the magnetic field frequencies and intensities that they use are consistent with the intensities and frequencies that you would expect to be driven by this type of quantum phenomena. So if this company is already making like almost $10 billion with this one magic magnetic field, imagine the power that you get if you can predictively change biology, not only for cancer, but for a whole other range of cellular functioning. So the technology to do this is already achievable. I mean, we don't need anything else than a cell phone. What's needed right now is this deterministic code book on how to go from the chemistry, right, to this physiological changes in a way that is more than looking for a needle in a haystack, right? Like trying different magnetic fields. We want to know deterministically how to turn those quantum knobs, so that maybe in a couple of years we can go to our cell phone and say, well, today I need help with wound healing. Wound healing is also something that changes as a function of magnetic field in a way consistent with this quantum stuff. And then you go to your app and say wound healing, and then your cell phone produces the exact magnetic field frequency and intensity that you need, and then you just apply your cell phone close to your skin or something like that. So that's the vision, right? But you need a more deterministic, mechanistic understanding of what's going on at the nanoscale in order to make this a reality.
Okay, so let me reflect on what we've talked about so far. So, you know, you have Niels Bohr and Einstein are contemporaries, and they're kind of competing against each other. And Einstein was always sort of didn't believe what Niels Bohr was doing. So, you know, they come by Einstein and Rosen and Podolsky come up with the CPR paper in 1935, and they're trying to disprove it. And in that paper, it suggests a quantum entanglement, and so that's like the ultimate flaw in quantum theory. And yet, as you indicated, transistors, electron
Right.
that occurs in
Yes.
semiconductors is based on quantum phenomena. GPS
Yes.
is based on quantum phenomena. Lasers are based on
Yeah.
quantum phenomena. So even though there's controversy and people don't fully understand it, it's being used and it's every.
It's being used. Exactly.
Right. So even here, there's something there. It does work, even though you may not understand all the pieces of it. If you can control it in a deterministic way. When you say deterministic, that means you can get a known outcome with
Yes.
a known trigger. Then you got something useful, just like the billions of transistors that are in my iPhone, or they
Absolutely.
all work.
Yes. Yes.
Or the lasers, or even some of the early work now that's occurring with quantum computing, and it's based on entanglement, right? Well,
Yes. Yes. Yes.
and work is coming out and saying, you know what, we've got quantum supremacy. It may be a toy problem, but it does work. Now let's see
Right.
if we can apply this, right, and get more coherence and
Right.
things like that. But what's exciting about your work is it can happen in squishy, wet environments and doesn't need superconducting and things like this, right? So
Exactly. That is what we want to unambiguously prove or disprove. Let me make a comment. People from quantum computing think that this comment is a heresy, but I'm going to make this comment anyway. I really think that quantum biology is today where quantum computing was like 30 years ago. In particular, 30 years ago, there was a lot of like theory done on
yeah.
like quantum protocols and stuff. And the first quantum gates were just started being implemented, right? Simple quantum gates with ions and stuff. So I think quantum biology is at this injunction where there's already a lot of theoretical quantum physicists modeling those things, how those things could happen. There's a lot of biologists and chemists doing those beautiful, hard experiments at those very distinct length scales. I think that the challenge now, I think the experiments that are really going to move the needle are the experiments that the quantum engineer can bring to this field. I think that with quantum-like experiments happening inside, say, a single cell, where you can initialize, manipulate, and read out, for example, those spin degrees of freedom inside a single cell and see how the preparation of certain spin states influence downstream physiological processes in a very controlled way. I think this type of experiment, like that the quantum engineer can do, I think that's the kind of experiment that is going to be necessary for the field to be accepted and to finally become mainstream, to have more than correlative but rather causal data.
Yeah, but you already indicated there's a startup company that's valued in the billions, and they've got something that is working based on this idea.
No, they don't say that. They don't say that it's based on this idea, and I don't think they are aware of it.
Right.
There's a lot of people. Actually, there's another company. There are two other companies from Singapore. One doing breast cancer treatment, the other one doing muscle regeneration.
Right.
All those fields are consistent with what would be expected in the range for this type of spin-dependent chemical reaction. In
Right.
particular, I don't think this mechanism from Novocure is not mentioned, and I think it's a consensus that nowadays you sort of find fields that work, which is, as I was saying, it's akin to finding a needle in a haystack because you have like a very big phase space. You have magnetic field
Right.
intensity, frequency, direction. Some fields are found that's good for particular things, right? For example, if you apply the magic Novocure magnetic field, you down-regulate cellular proliferation. But maybe a very close by magnetic field up-regulate cell proliferation, makes the cells, the cancer cells multiply faster. So it's all a guessing game right now.
Right. And you're trying to remove that guessing game, right? By doing the
Correct.
fundamental research to indicate the close tie-in between this foundational theory. And once you get this nailed, the foundational theory, and understand it, then you can more precisely control and create all sorts of
And
related
predict.
technologies that
Yes.
can be used. And that's where you're
Yes.
talking about the regulation of reactive oxygen, which is like
Yes.
oxidation versus antioxidants. If you can
Yes.
manage that,
Yes.
then you can do all sorts of treatments.
And again, there's data that this happens, right? There's data that exists for more than 40 years that weak magnetic fields change those things. It's
Right.
just heuristic, right? There
Right.
is no like, if we increase the field, this is what happens. If we change the frequency, this is what happens. So we want to put a little bit more systematizing into this. And I think
Right.
that the
Right.
way
Okay.
to do this is via quantum physics.
Right. And like you said, I mean, there's already there's evidence
Yes.
that there is this correlation or this or
Yes.
connection. It's more than correlation.
Yes.
It's like this connection between and causation, and you're just trying to figure out how to how do you fine tune these knots? But
Exactly. That's yeah.
yeah, you talked about oxygen species, but also epigenetic changes, which
Yes.
the environmental changes that can occur with gene expression in your body, and it could be even DNA methylene markers on your DNA.
Exactly. Yeah.
And if
Oh, embryogenesis. Oh, can I talk about embryogenesis? That's super
yeah,
cool.
absolutely.
So I have a friend who's a precision measurement physicist. What he does for a living is he builds very good Faraday cages, very good cages that shield external magnetic fields.
Mhm.
He does that for a living. He's like world expert in that. His name is Peter Fierlinger. He's at the TU Technical University of Munich. So the magnetic field of the Earth is about 50 microtesla. Inside one of his chambers, you have a noise level of about like 1 nanotesla. And then what he does is like he shoves ultra cold atoms experiments inside to do precision measurements. That's what he does. But then people started coming with ideas, oh, magnetic fields in biology. So here's what he did in his lab as a physicist. And now this has been reproduced. So he grew tadpoles, like frog embryos, inside those chambers under two conditions. In one condition, he applied inside a tiny magnetic field comparable to the Earth's, and after a couple of days, the tadpoles looked okay. In the other experiment, he grew the tadpoles for 2 days only under this very tiny nanotesla field. So after 2 days, only under this nanotesla field, 30 to 40% of the embryos were macroscopically deformed, macroscopically not viable anymore, like
Interesting.
wouldn't continue. Think about what this means, for example,
Right.
for space exploration. So you don't even need to add a field. Taking the tiny magnetic field of the Earth out seems to be doing stuff. And because the magnitude of the fields are so small, everyone's best guess is that it's not like a permanent magnet inside the frog embryos and things like this. And it happens in frog embryos, in other types of human cells. So, for example, can we reproduce in Mars? Can we do space farming in Mars? Mars has an ultra weak electromagnetic field.
Right.
Magnetic field. So I've been trying to talk to NASA people, right? It's not only about microgravity and cosmic radiation, it's also about magnetic fields.
Mhm.
Isn't this crazy?
Yeah, I mean, it's fascinating, and you know, you talk about this epigenetic changes to induce pluripotency. I think that's a really fascinating idea, right? So,
Yes.
can you describe that to the audience? What do you mean by inducing pluripotency?
So there's a lot of literature that shows that weak magnetic fields can change stem cells, right? So, for example, there are some animals or some organisms that are simpler than, say, a human that can regenerate very well. I mean, for example, from very simple planarians like little worms, flatworms, to like salamanders, they can regenerate very well. And again, I'm going to tone myself down here because I'm not trained as a biologist, but it seems that in such organisms that can regenerate limbs, they have a very high percentage of stem cells, like undifferentiated cells that can be deployed at any point to reconstruct stuff. With weak magnetic fields in certain organisms, you can actually change the pathways of of stem cells, like make more of one type of cells and less of the other type of cells. Imagine if we could do the same for humans again one day, right? By understanding exactly how those things work, it might be conceivable in the far future that once we lose a limb or something, that we can, in a very controlled way, put to use our own like very tiny quantity of stem cells to reconstruct this missing limb. Of course, this is complete science fiction right now, right? But I think we need to start doing research into like magnetic fields as applied to regeneration soon too. That's another very important aspect of this research.
Well, that's really interesting. It kind of reminds me of David Sinclair's or Yamanaka's, work in, you know, these four molecules. And
Tell me more. Tell me more. I don't know about that.
well, Yamanaka came up. In fact, he got the 2012 Nobel Prize for this,
Okay.
where you can apply these molecules to adult stem cells, and you can regress them into pluripotent stem cells. Adult cells, I should say. You can regress them to
Oh,
pluripotent
that's
stem cells.
super cool. Okay.
Yeah, and then what he did is he said, well, that's really interesting. I could sort of rejuvenate an adult
Yep.
cell into going backwards in time, and so
Oh,
it becomes a pluripotent stem cell. In fact, one that could be anything, right? And then he said, well, what if I stop that process somewhere between and let's apply it to a whole organism? So he applied it to, let's say, mice, and he was able to get them to rejuvenate, but then they would get cancer. They would get tumors, right? And then
okay.
maybe about a year ago, what they did is they found that not, I don't think it's his group, but another group found that if you don't use all four molecules, but you only use
Okay.
three molecules, they're called the Yamanaka factors. So you only use three and you carefully control them, you can actually do a rejuvenation without getting tumors.
Oh,
Cells out of
that's
control.
fascinating.
Yeah, so it looks like maybe you can apply this to different organs and rejuvenate. Now there's this idea of applying it to the whole body. And so in fact, Yamanaka has been he's embraced into a new startup which has massive
Ah,
funding. It's probably
cool.
trying to address this work in some way.
So cool.
But if you look at David Sinclair at Harvard, it's very similar. This idea of this epigenetic sort of uh, formula of some sort, and
Cool.
it carries some kind of information.
The,
And if you can control it, then you can get
does it require like genetic engineering?
it, from what I understand, and this is just one branch of the research, there'd be many applications. You can deliver it using probably some kind of safe virus to deliver
Oh, okay,
molecules, and
cool.
then you use an
Cool.
antibody to manage it, right? So,
Okay, cool.
and then the
No,
idea
that's
is,
super
yeah, and then rather than just applying it to one fragment of your body, why not try to do a whole body, right?
awesome.
But the idea then is if you can do that, then you can get a situation where somebody's or an animal that's older and you can regress them till they're like 24, the equivalent to OR 24, and then they start naturally aging. And so why at some point you maybe you can apply that to humans as well. So in my
Yeah.
keynotes, I always say, don't think of aging as mastering this idea as 50 years out or 100. It's
Yeah.
going to be the next 10
It's
years.
going to change. It's
Yeah.
going to change. And that's super
But your work is related, right? I mean, this
it is.
idea
So
of a weak magnetic
totally.
field that it can affect epigenetic changes, which then gets this idea into
Totally. And
uh,
and
pluripotency, right? So, and then
I think
yeah,
it's totally related, and it's a different way of thinking about how to actuate on biology, right?
right,
It goes from the molecular recognition, lock and key,
right,
from that to more like electromagnetics in biology, right? The cool thing about that, I think, and I again, I don't think I mentioned, is that for this electromagnetic stuff, there is no genetic engineering required
yeah,
because
exactly. It's
the knobs are
non-invasive.
there. Yes.
Yeah, no, I love it. I mean, it's like Mary Lou Jepson and her work that she's doing, right? Where she can get
Yeah.
brain cells to kind of implode just by non-invasive sound waves, or she can
Cool.
do other kinds of work with non-invasive near-infrared, I think, or some
Right.
kind of lasers,
Right.
but which penetrate the body, but in a harmless way, right?
Yeah,
But if
exactly.
you can do this with weak magnetic or magnetic fields, it's the same.
And
Yeah,
in some way, I think in the future it's going to be a combination of all those
right,
techniques, right? You can genetic engineer to have a stronger magnetic field response. I really think there's synergy there.
right,
And I think that electromagnetics in biology in particular has been underlooked,
right.
understudied, underestimated as this extra knob that we can use, as a control knob, really, right?
Right, and then you talk about cell proliferation and wound healing. So if you can somehow trigger
Yeah.
this the cell proliferation, then rather than the other side, if again if you can control the knob properly, you can control proliferation, which then helps with cancer.
Yes.
Or and on the other side, if you can go it the other way, then you can help with wound healing. I can see that.
Yeah, and you can help biomanufacturing, right?
Right.
Imagine if you can upregulate the production of lab-grown meat cells or bacteria that produces the enzymes or whatever that you care about. You make them multiply faster because of a weak magnetic field, or like
Yeah.
crappily produced by a cell phone-like device.
Yeah. Or cellular respiration rates. We talk about
Yeah,
ion channel functioning DNA repair. I mean, aging sometimes is thought of some kind of DNA problems.
exactly,
Yeah.
exactly.
So you talk about protein and growing proteins and things like this. The Terasaki Institute is working in this area. I should connect you to them,
Yes, I
or at
know them.
least show your work to them to see if they'd be interested. Philip Wong has developed at Stanford, and he's a semiconductor expert,
Yes.
but he's been able to reduce the size of these chips that they're so small they can be inserted into cells without disrupting the function of cells. What if you can
Oh,
use
that's
that?
oh they phagocyte the cells absorb the chips.
Yeah,
How does
yeah,
that run?
he's able to get, or he implants these chips into the cells, and they don't disrupt the function of the cells. They don't disrupt
Cool.
the ability of the cell to do its metabolism or anything else. Well, what you can do then is you can sense using these chips and control the behavior of the cells. But what if you were to integrate what you're doing with what he's doing? I think there's a kind of a synergy between his work.
That's very cool. That's very cool. I was not aware of that. I'm going to Google him. Cool. Nice.
Yeah, his name is Philip Wang, and because you know, transistors are, you know, they're familiar with electrons and spin states, and there's spin electronics now in quantum computing. This is not, I mean, it's not unfamiliar, right? It's familiar territory. So you should look, excuse me, look at his work, and then on the wound healing and cell proliferation side, and look at the work at the Terasaki Institute. They're actually at, just wait, you're at UCLA. They're at UCLA. They have a lab at
They're
UCLA.
very close to UCLA. They have a new campus now.
Yeah, they opened up a 50,000 square foot. Have you talked to them?
Yes, I have. Yes, I have.
Oh, okay, that's great.
They know me. They know of me. Yes, they know.
Okay, God, I mean,
isn't it crazy? I mean, there's a lot of synergy. I agree with that too.
And also with Philip and his work, right? So
Philip, I don't know personally, but that would be awesome.
Yeah, I'm telling you, it's actually really, really interesting, and I guess, like you say, this can get into drug development and therapeutic devices that are non-invasive, and you can remotely actuate them, and like you say, it's accessible as anybody with a mobile phone, right? I mean, it's amazing, right? And then the implications of space travel, and realizing that when you remove these magnetic fields, what is the implication?
Tiny magnetic fields, magnetic fields.
Yeah, exactly. I don't know why everybody should be looking at this.
I know. Oh, so you know what I heard? So here's what I heard. Because research in electromagnetics in biology is a taboo in the West, but research has never stopped in the East, like in China, Russia, and Japan. I was told by someone that probably one of the reasons for that is that after, again, not a historian here, historians in the audience, correct me if I'm wrong, but after World War II, the chemical industry in the West became super strong. The pharmaceutical industry, so that the cure for everything was chemical, in that there was a super strong pull to develop chemicals as treatments, right? So that might have pushed electromagnetics and biology a little bit aside, and maybe it's time to bring it back to the mainstream, right? As a very, in the end, Stephen, even if it's not a spin phenomenon that is driving those weak magnetic field phenomena, it's still a lever, it's still a knob.
Right.
We should understand how they work.
Right.
Even if it's not quantum, they work. I mean, there's evidence that it works. How on earth is that working? And if the spin theory doesn't work, we need to come up with another theory to understand this. The implications are the same. Your cell phone can treat you.
Yeah. So do you look at so you're an enthusiast, you're a researcher in this whole area of nanosensors and harnessing room temperature quantum effects on noisy environments. So it's quite amazing. But you got the support of UCLA, right? In this quantum biology tech lab.
Yes, I run my own lab. I've been funded by private foundations such as the Moore, Kavli, Idor Foundation, by NSF, also by the Army, the Navy, ONR.
Right.
There's a lot of military applications, you know, like
for sure, yeah.
For if you will, for bioelectromagnetic hazards, because if someone is trying to disrupt ourselves with an electromagnetic field, because the necessary fields are weak, you don't need a big weapon. You can use a small, you don't need a big cannon of electromagnetic energy or anything, because the necessary strengths are very, very weak. You just need to hit on that particular frequency and intensity, and you can either improve things or like improve performance, right? Or decrease physiological performance in ways that are not mapped out or that are not clearly mapped out for the world to see.
Yeah, I can see DARPA being interested in your work as well, right? Because the implications are so broad, but also for healing as well, right? So,
Yes, exactly. Exactly. It's a double-edged sword completely,
right.
right?
Have you talked to Hava Singelman? I think there's some.
Oh,
Hava
who's that?
Singelman has done work with, I think she's at Rutgers. She came out with a paper in about 92, 93 on super Turing. And it's this aspect of stochastic, out of the stochastic noise that comes out, you can get
Nice.
kind of these quantum effects that you can capture in analog and then get super Turing computation. And she's so transdisciplinary. The two of you just getting on a Zoom call or talk,
Awesome. That's I just Googled her. Cool. That's
you need to talk to her because
yeah.
I can see how you would have so much synergistic kind of more knobs to turn.
To turn to control. And interdisciplinary science is hard, right? Because
Right,
half of
right.
the time I don't know what I'm doing, right? But that's also the very fun part of it. We're always learning, and like everyone in the lab has a different, it has a different angle to contribute to the team. So I think that's very important too.
Okay, so I can see where your work came from. I can see where it's going, and now I'm going to ask you, what is the ideal case for you then? And let's say in 3 years' time, you know, you're working on this research, you're looking at the spin angle, but if it's not the spin angle, you're willing to pivot. You're just looking for something where you can understand it. I mean, because
The
you understand,
control,
yeah,
yes.
you can make it deterministic, right? So,
Yes, yes.
uh,
Right now, we already have more correlative data from our own lab and we chose parameters that were published in the literature, and we see the same things, which is good, right? Now we just need those big quantum-like machines to find out this code book, to go beyond those correlative data that uses magnetic fields from the literature or that make guesses of magnetic fields based on what was published before. And our goal is to, within 3 years, to have those big quantum-like machines. So what we build are like big uh, glorified microscopes with coils. They don't look like microscopes where you put your ear. They look like what you would expect if you opened the door for a quantum sensing, quantum computing lab. It's all synchronized down to the nanosecond. It's like a quantum experiment that works on a biological sample, if you will, right? And with those experiments that are completely sample agnostic and that can take data for more than a decade, as any other type of mainstream non-biological quantum experiment, we will be able, I think, to try to further the connection between those two length scales, the chemical length scales and the physiological length scales, by looking, for example, at the behavior of spins inside a single cell, inside proteins inside a single cell, for example, and see whether certain spin states are or are not correlated with some downstream physiological consequences.
did you know, the Global Good Lab got absorbed into the Gates Foundation, but prior to that, Mauricio Vecchione used to run it, right? So,
Who's that? Oh, tell me more. Who's Marija?
well, he's now the Chief Innovation Officer at the Terasaki Institute. His name is Mauricio Vecchione.
Oh, I know him. Okay, okay. Tell me more. I know Mauricio, but how
Well,
was he involved with the Gates Foundation before? Yes, tell me.
well, Bill set up a lab called the Global Good Lab and Fund. And what
Okay.
they would do is they would take concepts like what you have, and they would fund it totally to creating a solution, right? And that could be deployed globally, but it'd be the big picture kind of project they would take on. It's because when I toured the lab in 2018, I guess it was May, they showed me this new kind of microscope that they had created. They did ideation of it.
Okay.
Then they produced this first version that filled up an entire room. And then they showed me how they were able to iterate it down to something that was the size of a coffee pot.
Awesome.
And it's a new kind of microscope, right? But not in a
Nice.
non-traditional sense. And then they TerraPower and this liquid salt nuclear reactor, which is safe. They ideated the same thing, and then they were able to create it, and that's what they did. And they just constantly were doing ideation and taking something and totally converting it, translating it into something that could be used and scaled globally. And
that is so fascinating. Who's the contact there? Who do I talk to? That's fascinating.
well,
That's
if you know,
exactly
if
the type of, yes,
Mauricio would know. I mean, Mauricio would still. You should talk to Mauricio
cool.
because you know the people at Terasaki, right? So
Yes, yes,
talk to
that's
Mar
amazing.
Yeah, and
That's
say,
amazing.
Mauricio, ask him if the Global Good Lab within the Gates Foundation is still doing this kind of work, right? And if they are,
Cool.
this is something that I think, I think Bill Gates would be interested in this kind of work because the application is so broad, right?
I know. It's so underlooked.
Right.
It's there's a lot of potential there that should be explored. And in the end, quantum or not, it's a knob. It's a
It's a
weird
knob.
knob.
Yeah, exactly. So
It's a
let's
weird,
understand the knobs better, right?
yeah, yeah, exactly, right. It's our understanding of biology. So right now, biology is sort of at the Newton's laws level, right? How mechanobiology, if you push something, what happens? This is like really, at least if not quantum, if not light matter interactions as we would call in the quantum community, it's at least electromagnetics in biology. So
Right.
we need to move out further than Newton's laws and go to Maxwell's equations in biology, right?
Right.
And then from there maybe to quantum. Yes, quantum, no quantum, but yeah, it's a knob.
Yeah, and I mean, one of the scientists that they work with at the Teraseki Institute, Shiling Shen, and
I
he,
know, Sheila. He's awesome. He's so
yeah, I
nice.
know,
He's
and
so
he's got
nice.
electrical engineering too, right? So
I know, I know, I know, I know. And when I talked to him, I think he got what our vision was. He's
yeah, he
awesome. He's
would for
awesome.
sure, right? And because you're both electrical engineers by training, then you want to look at the practical side of this thing, and you
Yes,
understand
yes,
the quantum phenomena because electrical engineering is sort of based on
yes,
quantum phenomena, right? So
yes.
okay, well, we're down to our last question, and sort of what would you like to leave the audience? What sort of recommendation?
Well, first of all, thank you for having me. It's been a pleasure talking. As any scientist, I love talking about my research. Thanks for humoring me. Well, two things I want to share with your audience. The first is about quantum literacy. In the same way that 25 years ago, the only people who learned how to code were maybe engineering or physics undergrads, and now the educationally privileged are learning coding even as early as middle school, and every field of knowledge benefits from knowing how to code. Think about even linguists coding for speech patterns, right? Coding is recognized as a universal skill. I hope and I think that the same will be true for quantum mechanics, the instruction literacy in the near future, because quantum mechanics is required again to understand the world that you already live in,
Mhm.
right? To understand how basic things such as your classical computer, your laser pointer, how those things work. So I hope that quantum literacy becomes a thing, becomes recognized as a universal skill set. And I think that's also the way that we're going to end up bringing more people to the game of quantum biology. If everyone, chemists, biologists, engineers, material scientists, if everyone has an idea about what quantum is and the power of quantum, I think that there's potential for us to discover what quantum biology can bring. The second thing that I would like to leave your audience with is the I'm very proud of that, but the American Physical Society has just published my opinion piece called "It's Time to Take Quantum Biology Research Seriously." I'm very happy because this is one of the first times that the American Physical Society actually recognizes this field. And I think it's an important path towards validation of what we're trying to do and understanding of what it takes to do interdisciplinary research and how far can it get us, right? To the idea of like being healed by your phone at some point in the near term.
Thank you so much. And Clarice, you got to make sure you send that a revised profile for the interview
I
with
will.
all the links,
I
including
will.
to your work at the lab and so on, right? So,
I will. I will.
and Clarice,
Thank
it's
you.
such a pleasure to have you come in. I really enjoyed this
Me too. Thank you for
forward to
humoring me.
chat with you. So,
Thank you for teaching me the things about biology and stuff like those small molecule stuff. I'm going to read more about that too. Always an opportunity to learn.
so take care and thank you again.
Thank you. Talk to you, Stephen. Thank you so much.
Thank you for listening to the Brand Called You videocast and podcast, a platform that brings you knowledge, experience, and wisdom of hundreds of successful individuals from around the world. Do visit our website www.tbcy.in to watch and listen to the stories of many more individuals. You can also follow us on YouTube, Facebook, Instagram, and Twitter. Just search for the Brand Called You.